Good afternoon, everyone. Hello. Just waiting for some people to join. I see the numbers coming in. Okay, good afternoon, everyone, and welcome. My name is Jocelyn Badali, and I am the research lead at the Alzheimer Society of Canada in the Research, Knowledge, Translation, and Exchange team. The Alzheimer Society of Canada is very pleased to welcome you to the second ASRP Exchange webinar that is being offered through the Provincial Support Program. This series has been developed to feature Alzheimer Society Research Program recipients who have completed their funding and who will speak to the outcomes of their ASRP supported research while addressing how their results will continue to impact people with lived experience. So with me today, I have Caitlin Jaggers. She is the Research Program Assistant at the Alzheimer Society of Canada, and she will join me today in moderating our program. And in addition, we're also pleased to have Noah Ben-Gaed, the Director of Research from the Fédération des Sociétés Alzheimer du Québec, joining us today as co-host. So I'm just going to pass it over now to Caitlin as she will go over some of our housekeeping tips for the webinar today.
Hello, everyone. My name is Caitlin. As Jocelyn mentioned, I am the Research Program Assistant at the Alzheimer Society of Canada within the Research, Knowledge, Translation, and Exchange team. And I will start off today's webinar by going through the agenda presented here, and then we will pass it over to our presenter, Dr. Almey. Before we begin, please note that this webinar is estimated to take under an hour to complete, and it is being recorded, but that only the video and audio of the presenters is captured during the recording. The presentation recording, in addition to the PowerPoint slides in PDF form, will be made available on connection following today's webinar. Today's webinar is presented by Dr. Almey and is being offered as one of the ASRP Exchange webinars within our new provincial support program, Meet the Researchers series. We are so pleased to have Dr. Almey with us today to present her project, Early Loss of Ovarian Hormones Negatively Impacts Cognition and the Frontal Cortex. This project was funded by the Alzheimer Society Research Program, the ASRP, as part of the Postdoctoral Fellowship in Women's Brain Health and Aging. This started in 2017, with project completion ending in the fall of 2019. Dr. Almey is a research associate with the University of Toronto and the University of McGill, as well as an assistant professor in the psychology department at McGill. Anne works for Dr. Gillian Einstein's lab at the University of Toronto to establish a research site in Montreal. The project she is working on examines how loss of estrogen following removal of the ovaries contributes to cognitive decline and changes in the brain, and asks whether hormone therapy following ovary removal can protect against these changes. With the assistance of our co-host Noah, we will be moderating a short question and answer period following today's presentation. Please type any questions you have into the chat box if you have any comments you would like to make or questions you would like to pose during the presentation. However, please note we will hold off on asking these questions to our presenter until the presentation has completed. Without further ado, please join me in welcoming Dr. Almey.
Okay, hi guys, just give me a second and I'll get my screen shared and we'll get going here. Okay, I think we are good to go.
Perfect.
Go. Sorry, I'm just hiding all your videos on Zoom so I can see what I'm doing. So thank you very much for such a nice introduction. I'm really glad to be here today to speak with everyone, tell you about my research over the past couple of years, both because it's really exciting to share the results of my work, and also as a thank you to the Alzheimer Society of Canada for contributing so generously to the fellowship that really did make it possible for me to do this research. So since I really started doing research during my undergraduate degree at the University of Guelph, my interests have focused on ovarian hormones. So I'm sure many people know this, but ovarian hormones refer to a couple different groups of hormones. So first we have estrogens, and Fun fact, estrogens actually are not a hormone, they're a group of hormones, including estradiol, which I'll talk about a little bit today, and also estrone and estriol. And then the second ovarian hormone or group, I guess, is progesterone. So my research interests over the past 8 years have focused on how these hormones affect the way that females think and behave, and how these hormones might contribute to health and disease in females. So you can see that that's carried into the project I'm going to tell you about today, since I'm talking about how loss of these hormones might negatively impact both cognition and brain structure, specifically the cerebral cortex. Now, this work fits into the mandate of the Alzheimer Society of Canada because any changes we see related to ovarian hormones might affect the risk for Alzheimer's disease. So it turns out there is a significant sex difference in Alzheimer's disease. So according to the most recent Canadian statistics I found, which were from 2017, 63% of the cases of Alzheimer's disease in Canada are in women. And that sex difference is observed in European cohorts and Asian cohorts as well. So it seems pretty consistent. And I'm sure many of you are familiar with the fact that Alzheimer's disease is predicted to rise over the next couple of decades as our population ages, increasing the burden on the Canadian healthcare system. So I've got a nice visual here from the Alzheimer Society of Canada, just showing the predicted increase in Alzheimer's and the cost of that increase. But of real interest to me is the fact that it's predicted that this sex difference will continue to grow. So by 2031, it's estimated that 72% of cases of Alzheimer's will be in women. So women are really bearing the burden of this disease more so than men. Now, in addition to there being a higher rate of Alzheimer's in women, women also have more severe symptomatology. So women at the same stage of disease progression of men have more severe cognitive impairments across a number of domains. So things like verbal memory, visual spatial memory, episodic memory are all more severely affected in women. And recent work from the Alzheimer's Disease Neuroimaging Initiative suggests that once women are diagnosed with mild cognitive impairment, that sort of precursor stage to Alzheimer's disease in a number of cases, that women decline almost twice as fast as men. So not only are women diagnosed more frequently than men, but when they are diagnosed with Alzheimer's, they have more severe symptoms and more rapid decline. So in a broad sense, what my work was really trying to address is why this is the case. Why do we see this sex difference? Because if we better understand why women are at this elevated risk, it could let us develop interventions at specific time points in life or targeting specific lifestyle factors that could delay or possibly even prevent Alzheimer's in this at-risk group. So one group of women that's at particular risk for Alzheimer's disease are women who undergo a surgery called oophorectomy prior to natural menopause, which typically occurs in women at about 50 years, which is the cessation of menstrual cycles. So if women undergo this oophorectomy surgery, and here I've got a picture of female internal reproductive organs. We've got the uterus, the fallopian tubes, and the ovaries here. Oophorectomy refers to surgical removal of the ovaries. So if women's ovaries are removed before they would naturally undergo menopause, this almost doubles their risk of Alzheimer's disease. Which is really kind of incredible because women are already at elevated risk even without this surgery. Now, the studies that show this are primarily epidemiological studies. So they look at data on women from hospital records or Medicare records in the States, that sort of thing, and noted, okay, women who have this surgery are at risk. And they also noticed that if women undergo this surgery but are prescribed hormone replacement or hormone therapy, so ovarian hormones to replace those that are lost when you take the ovaries out, this seems to reduce their risk of Alzheimer's disease. So a great question here would be, why would women undergo this surgery if it puts you at such an increased risk for late onset Alzheimer's? And there's a few reasons for that. But the reason I'm going to focus on right now, because it's pertinent to my research I'm going to tell you about today, is women who carry what's known as the BRCA1 and BRCA2 genetic mutations. So BRCA stands for breast cancer, and women carrying these mutations are at significantly higher risk of developing breast or ovarian cancer. And so current best medical practice, what's recommended, is that these women have what's known as prophylactic bilateral salpingo-oophorectomy. That's a mouthful. I'm going to shorten that down to BSO for the rest of the talk. And what that really means is it's a risk-reducing surgery where these women have both their ovaries and the fallopian tubes removed on both sides of the body. So these sort of purple dotted lines here, at approximately the age of 40 is what's recommended for them right now. So about 10 years before natural menopause occurs, which means that these women represent a group with reduced exposure to ovarian hormones, early loss of ovarian hormones from this surgery, who are at this elevated risk of Alzheimer's. Now, epidemiological studies have shown us that they're at this elevated risk, but what my work wanted to tap into is a little different. So we wanted to look at women shortly following their BSO surgery to see whether their memory or their brain structure are affected, in the hopes that this would give us a better idea of the time course of changes that might be occurring in response to this surgery. So I was lucky enough, all of the work for this postdoctoral fellowship was done in Dr. Gillian Einstein's lab. That's her up there. She's the supervisor for my postdoctoral fellowship. And I was really lucky because the Einstein lab had started this project well before I joined the lab. So I joined a lab with the project up and running and a lot of data collected, which really made it easy to run the analyses I'm going to tell you about. So the Einstein lab, even before I joined, had been recruiting women carrying this BRCA1 or 2 mutation who had undergone BSO surgery in Toronto, because Dr. Einstein's lab is at the University of Toronto. And she'd also extended the project to Linköping, Sweden as well. And my role as her postdoc, I'm actually in Montreal, not in Toronto. I set up a Montreal arm of this study. So it just expanded the study into a third city. You can imagine that women with these genetic mutations who undergo this surgery are relatively rare. So by expanding where we could recruit women from, it makes it easier to get people in the study and get the sort of sample sizes and results that we'd like. So as I said, Dr. Einstein was recruiting women who'd undergone BSO. And she accepted women who'd undergone BSO and were not prescribed hormone therapy following this surgery, and also women who'd undergone BSO and were prescribed hormone therapy after their surgery. In addition to that, the Einstein lab also was recruiting control participants, so women of a similar age to the women in the 2 BSO groups who had not undergone any surgery, had no known problems with reproductive function, etc., so that you could make a comparison between controls and women who'd undergone BSO to get an idea of the effect of this surgery. Now, the study being run out of the Einstein lab is really huge and has a ton of potential because they are collecting a bunch of data I'm not going to be talking to you about today, but I wanted to let you know what the whole study looked like. So data is being collected on a number of different cognitive processes. I think there's 18 different sort of aspects of cognition being tested, but data is also being collected on sleep, on stress, measures of mood like anxiety and depression. And more recently, the study started looking at brain structure using MRI scans and brain function using functional MRI scans. And this is a really exciting data set because it's looking at a lot of different variables or factors that we think might be affected by loss of ovarian hormones, and also factors that are known to contribute to Alzheimer's disease risk. So this really has the potential to give us a good idea of how changes in a number of different things following loss of ovarian hormones might contribute to the increased risk of Alzheimer's disease that we know is occurring for women who undergo BSO surgery. And even more broadly, like definitely the results of this study are going to speak to women who undergo this surgery, but more broadly, all women undergo a loss of ovarian hormones at natural menopause, as I said, around the age of 50 years old. So it's possible that the menopause that every woman goes through might be contributing to the general increase in risk for Alzheimer's in women, and that some of our results in this study can help to understand Alzheimer's risk in women in general. This study really provides the opportunity to tease apart the effects of ovarian hormone loss and aging. So that is one of the things we're really hoping can be a takeaway from the results of this study in general. Now, more specifically, we're going to get into the actual experiment that I ran, because I definitely didn't have time to look at all the data that was collected in the Einstein Lab study. So my specific series of experiments looked at how BSO surgery affects a couple types of memory and one aspect of brain structure known as cortical thickness, which I'm going to explain in a minute. So I went through all of the women that had been recruited to the Einstein Lab study so far, and I pulled out women from Toronto, from Linköping, and from Montreal who had undergone MRI scans so that I was able to look at brain structure, this measure of cortical thickness. And so what you can see here, my three little color-coded groups, is that I ended up with 21 women who'd undergone that BSO surgery and were not taking hormone therapy, the BSO no HT group. I had 21 women who'd undergone BSO surgery and were taking hormone therapy. And then I selected 42 controls that were of very similar age to the women in my BSO groups to try to make sure that age wasn't contributing to any effect I saw. We were really looking at the effect of losing ovarian hormones. Now, as I might have mentioned on the previous slide, but the women that we were recruiting here are relatively young. So the average age of all the women in my study was 42 years. So we're looking at early middle-aged women within 5 years of their surgery, so relatively shortly following loss of ovarian hormones. And I also wanted to point out here that women in the BSO-HT group were taking a sort of mixed bag of hormone therapies. So their doctor was prescribing what they felt was best. And as a result, the women were taking some different hormones or different methods of administration, a little bit of different dosing. But the commonality between all women in these groups was that they were all taking a specific form of estrogen known as estradiol, designed to mimic levels that are seen across the natural menstrual cycle. So in terms of the measures of memory I was looking at, first I wanted to look at the working memory performance of women in this study. And to do that, I used a digit span task. And there was sort of an easier and a harder version of the digit span task. So in a digit span task, you read a series of digits out loud to the participant. And for the easy version, you ask them to repeat back those digits to you in the same order. The more challenging version of the task, you ask them to repeat back the digits to you in reverse order. And I also wanted to look at verbal memory. And to do that, I used data from the logical memory task. This task, you read the participant a short story, 3 or 4 sentences, and immediately following the story, you ask her to tell you any details that she can remember about the story. And then you go on with the cognitive testing session, and 30 minutes later you say, do you remember that story I read to you a little while back? Now tell me all the details you remember again. So the easy version is the immediate logical memory, and the harder version is delayed. Now, as I said, I also wanted to look at a measure of brain structure called cortical thickness. So I was using what are known as T1 weighted images from an MRI scan. And you can see an example of that here. It really looks like a photograph of the brain inside of the skull. And the cerebral cortex is the sort of wrinkled area outside, around the outside of the human brain. So I hope you can see my pointer here. And cortical thickness really refers to the gray area that wraps around the very outside of the cortex. So I used a software called Civet, which wraps a surface around the white matter, sort of inside the cortex. You can see that in the yellow line here, and then wraps a surface, the red line, around the outside of the gray matter. And so cortical thickness is the distance between that yellow and red line. So how thick that gray matter is around the outside of the cerebral cortex. So I took these measures of memory and this measure of cortical thickness, and I compared it between the BSO no HT group and controls and the BSO HT group and controls to look at the effect of BSO surgery on these measures of memory and brain structure. And then I also compared the 2 BSO groups to get an idea of the effect of hormone therapy. So I'm going to get into my results now. Spoiler alert, I feel like that title for the slide definitely spoils the upcoming graph. So we did observe poor memory performance following BSO. but there's some caveats to that, so I'm going to get into that now. Now, in our working memory task, the digit span task, first of all, when we looked at the easier version of the task, digit span forwards, we didn't see any differences between groups, suggesting that on this easier version of the working memory task, all of the women in our different groups performed similarly. However, when we looked at results from the more challenging digit span backwards task, what we saw, so on the x-axis here, the y-axis here, we see the total number of digits the women remembered, and you can see that women in the BSO no HT group remembered significantly fewer digits than controls. And what you see here when it comes to the BSO HT group, the women taking hormone therapy, is they were not significantly worse than controls, but they were not significantly better than the BSO no HT group. So they were sort of sitting somewhere in the middle between these other two groups. Now, when it comes to the verbal memory results, we saw almost identical things. So on the easier version of this task, the logical memory immediate, there were no differences between the three groups. So it looks like they were performing equally well on the sort of simpler version of the verbal memory task. However, when we look at the delayed version of this task, we see a very similar pattern of results, where women in the BSO no HT group remember significantly fewer details about the story than control participants. And women in the BSO HT group were somewhere in between. So they weren't significantly worse than controls, but not significantly better than the BSO no HT group. So just to sort of summarize all of this business, women who underwent BSO surgery and were not prescribed hormone therapy had poorer working and verbal memory than control participants, suggesting that maybe BSO might be leading to some of these memory deficits. And while women who'd undergone BSO and were taking hormone therapy following this surgery, their memory was not significantly worse from control participants, but they also were not significantly better than women who underwent BSO and didn't take hormone therapy. So they were sort of somewhere between the two groups on both measures of memory. Now we're going to talk about that in a little more detail, but first I wanted to show you the results of the cortical thickness analyses. So as you can see here, we're looking at a brain from a number of different angles, just so you can see all of the different parts of the brain. And in this image, we've got some areas showing up in turquoise. Now, what those areas depict are regions in the BSO no HT group that were significantly thinner than control participants. So we saw areas in the right frontal cortex, specifically the dorsolateral frontal cortex and the frontal pole right at the tip of the brain here, right up front, were thinner in women who had undergone BSO surgery and were not prescribed hormone therapy. Again, similar to the cognitive results that we saw, women who underwent BSO but were taking hormone replacement of some sort did not have any areas of the cortex that were significantly thinner than controls, but they didn't have any areas thicker than women in the BSO no HT group. So again, it seems like these women were sitting somewhere in the middle between control participants and the BSO no HT group on measures of cortical thickness as well. So I'm just going to summarize the main results I told you about today, and then we can get into a little discussion if you guys have any questions. So really the main finding, the take-home message, is that relatively shortly following BSO surgery in young middle-aged women in their early 40s, women are exhibiting poor performance on both working and verbal memory tasks and have thinning in the right frontal cortex. So I wanted to point out here that when it came to the memory performance, these women were only exhibiting poor performance on the more challenging versions of the working and verbal memory tasks. So this suggests that we're seeing very subtle changes in memory. If you give them an easy memory task, they perform just like women who haven't had the BSO surgery. But if you give them a more challenging version of the task, that's when we start to see deficits. So it's a subtle memory change we're seeing at this point. And that really makes sense. These are young women. Most of the women were busy raising families, had jobs, were very cognitively active. So we wouldn't expect major memory differences or major changes in the brain. We only saw thinning in one area of the frontal cortex. But these subtle changes occurring at midlife, you can imagine 15 or 20 years down the line, if these sort of changes continue, how they might contribute to these women being at higher risk for Alzheimer's disease. Now, we thought these results were really interesting because the frontal cortex is important for working and verbal memory. So it's possible that the changes we see in the cortical thickness, the thinning in the right frontal cortical thickness, could be contributing to changes in memory performance. And we also thought it was interesting because there's specific patterns of cortical thinning that are observed in Alzheimer's disease. And these patterns include thinning in the dorsolateral frontal cortex, which is what we saw in the women with BSO not taking hormone therapy. So it really does make a link between these potential very early changes in the brain following BSO and the later increased risk for late onset Alzheimer's disease. Now, another take home from our study is that hormone therapy seems to mitigate the declines in memory and cortical thickness that may be occurring following BSO. And as you might remember, our women were taking a sort of mixed bag of hormone therapies, but since they were all taking estradiol replacement specifically, we hypothesized that there might be some protective role for estradiol replacement in terms of reducing changes in memory and the brain following BSO surgery. But I would also note here that the women in the BSO HT group were always sort of in between controls and women who'd undergone BSO without hormone therapy, suggesting that this hormone therapy they were taking helped reduce changes, but maybe didn't eliminate them. So hormone therapy is helping, but maybe not as much as maintaining a natural menstrual cycle. So sort of to wrap up my talk before we get into the discussion, it seems that this early loss of ovarian hormones and potentially loss of ovarian hormones in general that can occur at natural menopause could contribute to declines in both the cortex and in memory that may precede later life Alzheimer's disease. So I'm happy to discuss any of this. I glossed over a lot of the scientific nitty-gritty and a lot of the background because I didn't want to take too long talking. I wanted to leave time for the discussion. But before we get to that, I just wanted to thank a few people. So I wanted to say thank you to my supervisor, Dr. Gillian Einstein, and the entire Einstein lab and the lab in Linköping, Sweden as well, who helped collect the data that I've presented to you today. And I also wanted to thank Dr. Natasha Raja and the Raja lab, who are here in Montreal, and they acted as my home base as I was setting up this study, the Montreal test arm for this study. I also obviously want to thank the Alzheimer Society of Canada for contributing to my postdoctoral fellowship, as well as the Ontario Brain Institute, the Women's Brain Health Initiative, and CIHR, who contributed to my fellowship and the money to do this research itself, and a couple of the Swedish funders who allowed for the research to be done in Linköping. So thanks very much for everyone who's here, and I'm looking forward to talking about this work in more detail.
Thank you so much, Anne. That was wonderful. And we really appreciate how you explained it in lay terms to some of us who are unfamiliar with this kind of research. So that was greatly appreciated. I noticed that we have a question over in the Q&A, so I'm going to hand it over to Noah to start us off with that.
Thank you, Jocelyn. So we had indeed a couple of questions who came in during the presentation. The higher prevalence and incidence of Alzheimer's disease in women remains debated. What are your thoughts on the fact that these differences have been observed in European cohorts, but not so much in American cohorts? Are they confounds here? And if you could discuss this issue?
Yeah, for sure. So that is definitely true. This sex difference in Alzheimer's disease seems pretty consistent in many cohorts that I've seen data on, except American, where it occurs a little bit less. It seems like the rate of Alzheimer's might be higher, but the prevalence and incidence of Alzheimer's in studies done in the United States seems to be much closer between men and women. So it is actually a little bit, it's pretty hotly debated in the very niche world of research that I work in, why this might be the case. And I have to honestly say, I don't think anyone has a complete answer, but there are a number of factors that could contribute. So I think the cohorts of women being examined, or the cohorts of people being examined, might really be important. So as we get studies in younger groups, where women are more educated, more likely to have had an occupation that keeps them thinking into their old age, maybe are more likely to be physically active and stuff like that. Those things we know all protect against the onset of Alzheimer's disease. So it may be that some of those factors, that women started participating in those a little bit earlier in America, or that women who participate in those types of activities are more likely to take part in studies in America, which may contribute. It may have something to do with other health factors. So I know that diabetes is higher in North America. It's not my main area of study, but then Europe as well. So it may be some other health factors in the United States, North America in general, differ between places, and that could be contributing as well.
So another question came in as well. Is there any research or application of this data to extended 5+ years of amenorrhea and those who prematurely lost their period at a younger age?
So any studies, sorry, what was the first one?
So about research and application for on data to 5 years or more extended amenorrhea.
Okay, so I do see what you mean. And in fact, recently, I think a lot of the results that I'm going to talk about in a second were published while I was doing my research. But they really bolstered our confidence in our findings, because women who lose their ovarian hormones before natural menopause because of this surgery I was discussing during my presentation are at higher risk. But there's now a couple different studies from sort of separate research groups, so people are finding this independently, that the duration of ovarian hormone exposure predicts Alzheimer's disease. So if you get your period later in life, or if you undergo natural menopause earlier in life, sort of shortening that window that a woman is exposed to ovarian hormones, because that is during the time that you have menstrual cycles from the onset of puberty until menopause. If you have a shorter window of exposure, you're at a higher risk of Alzheimer's disease. So early or premature menopause definitely does increase risk in a similar way to BSO. And I would expect that, I don't know if I've seen a study on 5 years plus of amenorrhea, so loss of menstruation sort of during the midlife period before menopause should have occurred. But my educated guess would be that that might have a similar effect. And I don't know of any clinical trials specifically in premature women who've undergone premature menopause, but it's possible that hormone therapy might be beneficial for them in the same way that it seems to be beneficial for BSO, although we still have research to do before we feel really confident about that.
Another question as well on the same topic, and I guess you also already answered it. What are the effects of removal ovaries have on cognition on women under 40s? And has there been any studies to follow them to see if they experience cognitive decline approaching 40 or 50s?
To the best of my knowledge, like before 40, so some of the women in our sample did have their ovaries out before 40. There definitely was a range. And I think the earlier, I think the earlier you lose your ovarian hormones has potential to have greater effects, just because if that instigates changes, there's a longer period of time over which detrimental effects could occur. But I don't know of any studies that have really tracked women following this surgery. So a lot of the evidence comes from looking at women who have mild cognitive impairment or Alzheimer's and saying, hey, a larger proportion of these women have had loss of ovarian hormones or have undergone this ovary removal surgery. Maybe it's contributing. Tracking it following surgery is is pretty rare in the field of research. And some work has been done actually out of McGill University. Barbara Sherwin did a lot of this work in the early 2000s, but she tracked women for a relatively short period of time. So within 6 months of their surgery, she looked at memory and how replacing estradiol can affect that memory, and saw declines in verbal memory, and that estradiol protected against this. But that leaves us with a huge window, right? Between 6 months after surgery and 65 years of age is a big window that's a bit of a black box, which is some of what the Einstein Lab is trying to understand better.
You mentioned hormones that have an impact on cognitive decline, but is there any research to determine if one of the hormones play a protective role, and maybe instead of undermining the conditions of women after an ovarian vasectomy to really have an impact on a protection, because apparently there is multiple hormones that contribute to estrogen being protective factors?
Yeah, so I would say there's evidence suggesting that if there is a protective factor of one of the ovarian hormones, it's most likely to be estradiol, that estrogen that I was talking about during my presentation. And there's evidence from rodent work and primate work that really suggests this. The evidence in humans is a little bit more challenging. And that's partly because often in studies like ours, women are prescribed whatever their doctor recommends. So we don't get as clear a picture because women are taking different doses, different methods of administration. But estradiol is the main estrogen produced by the ovaries in naturally cycling women. So women who are, mid-age and still menstruating. And that hormone declines almost entirely. It's almost lost following menopause and following the loss of the ovaries. So based on animal work and some human work, and the fact that that is the hormone loss, we suspect estradiol is playing an important role. But there are some caveats. So it looks like estrogen is really a potential here, but some past studies have shown risks of hormone replacement, not really with estradiol specifically, with other formulations of estrogen, but it's why we really need more carefully controlled clinical trials to ensure that hormone replacement we want to give is safe and to make sure it's having the effects we think it's having. We need women to be really taking similar doses of the same hormone for the same amount of time so we feel more confident in the results in humans.
I think there is probably one open question that we received a suggestion for the KT&E research group to answer it. If they would like to take this opportunity and probably add some background to it.
Yeah, certainly. So I just, I noticed there's a question. I noticed there's a chat question from someone who's just potentially interested in reaching out to you, Anne, so we can talk about that afterwards. Yes. I just have a couple of questions on behalf of the Alzheimer Society. We wanted to just ask you a little bit about your experience holding this fellowship. So you essentially were working with 2 research institutions, as you had mentioned. So we're wondering if maybe you could speak a little bit about your experience working with the 2 supervisors and how the funding through this ASRP Postdoctoral Fellowship has sort of impacted both of these investigative teams that you worked with in order to accomplish your research goals.
Yeah, so it was really a unique postdoc because I was sort of divided between 2 cities. I am in Montreal and I've done all my work out of Montreal, but was back and forth a little bit. And the Alzheimer Society postdoc really, first of all, it provided the funding so that I had a salary so I could live while I was doing this research. It also provided a small amount of money that allowed for my travel between Montreal and Toronto, which meant that I could work in Montreal, but still regularly connect with the research team in Toronto, which is really important. It's nice to be able to see, to really sit down and meet with the people doing the same work as you. And in general, I would say that receiving this fellowship just let me do work that was kind of a dream. During my PhD, I did animal research, and I've transitioned to human work for this postdoc. And in animals, often, if you want to look at the effects of estrogen, you remove the ovaries from a female rat, and that gives you an idea of, if you're looking at the difference between a rat with no ovaries and a rat with ovaries, what are the ovarian hormones doing? And we always sort of joked a bit, but this doesn't really apply to women. So this postdoc was a dream for me, because this was like a human model of what I'd been studying in animals, and a way of taking all of my doctoral work and translating it into humans, which was something I was really excited to do, because that's what we always want to. I was doing the rat research to better understand people. I like rats, but not that much. So this postdoc really gave me the opportunity to make that leap into humans and to work with such an interesting population of women. I really hope that the results of this research can benefit those women.
That's fantastic. And another question with respect to holding this postdoctoral fellowship, what perhaps advice, like whether it's career or academic, would you give to individuals who are pursuing a postdoctoral research position with respect to what's going on with COVID-19 right now? Because you had mentioned your ability to travel. So I'm wondering if you have any comments about that for the future.
Yeah, so the pandemic has not made anything easier for anyone, I don't think. And that's true for research as well. So particularly, I think animal research, there's been a lot of restrictions with being able to do research in general, but doing research with people is really a challenge right now, because it's very difficult to invite them into the hospital where I did most of my research out of. It's not safe for them. It's not that safe for researchers to be there, or for people in the hospital to have us in and out and in and out. So it's really restricted the research that can be done. So my advice would be, get creative. Look for data sets that already exist. If there was a question you wanted to ask, maybe you can find a data bank that you can tap into and apply some of your questions to that while you're waiting to be able to get back into research. And I would also say, look to online research. Obviously, we're all figuring out ways to do stuff online we didn't used to, since we're all here today. And I know that that's been really a direction that the Einstein Lab has gone in. So we tried to transition a bunch of testing to online, so that we're able to reach out to people and continue to do research, even though we can't meet with them in person. And also use this time to get your papers written. Nobody wants to write up all the data, but it has to get done. It's a good opportunity to do that as well.
Probably a question more related to the study you presented today. What was approximately the long post-surgery when the women were in the study? And if I can add as well to this question, for how long women were under hormone therapy?
Yeah, good questions, because all of that is definitely going to affect the results. And as you know from my talk, it was a little bit of a mixed group, because we were recruiting women as we met them. But on average, although there was a range, the women in this study were 5 years out from the BSO surgery. So that definitely varied, but all women underwent the surgery before they went through natural menopause, and they were on average 5 years from surgery when we tested them on their cognition and acquired their MRI scans. And then the duration of hormone therapy often varied with the length from surgery. So we noticed, even Dr. Einstein would say, across the time that she's been studying this group of women from 2013 until now, many more women are being prescribed hormone therapy when their doctor feels that it's a safe option for them, which seems great, because our results suggest it might be beneficial for them. And other results, there's other studies that suggest a number of other benefits of hormone replacement outside of cognition in the brain, which is my wheelhouse. But so the women had typically been on the hormone therapy since their surgery. So it varied. Often the doctor will prescribe hormones 3 to 6 months following therapy or following surgery, and they'll continue to take those hormones until approximately the age of natural menopause. So they're early to mid 50s.
Okay. You mentioned in the study that you only focused on people who had the surgery and who had the BRCA1 and BRCA2 mutation.
Mm-hmm.
Do you think that your result can be applied to women who would have lost their ovary for other reason?
We think so. So that's another good point, right? The women that had underwent BSO surgery were potentially different from other women in that they carried this genetic mutation. To the best of our knowledge, this mutation mostly affects cancer risk. It doesn't seem to have any cognitive impact. And in our data set, we've compared, there's another aspect of the study that I didn't even talk about. It's too big to get into all the details, but we are looking at women who carry that mutation and are considering having the surgery, haven't had it yet, with the hopes of testing them before and after the surgery. So we have a group of women who carry the mutation and have not had the surgery. And we did compare them to controls, and we don't see any sort of cognitive differences or anything in women who carry this mutation. So we think differences we're seeing are based on the surgery, not based on that mutation. And that means that we're really hoping the results do generalize to women in general, not just women with the BRCA1 and 2 mutation.
There is a very good comment on your study and your presentation. Thank you so much. A question that came in was there any variability in cognitive performance based on race or language, or the preference or the socioeconomic status of the women who were involved in the study?
So that is a really good question. Intersectionality is becoming such a popular topic as we realize how those factors influence our results. And I have to say, we did not look at that. Now, the other thing I will say is our population was predominantly Caucasian. Most of the women we studied were Caucasian, although there were some exceptions to that for sure. And I would say most of the women we studied were middle class, just based on level of education and occupation, the sort of things we had them report in demographics. But we didn't look at how those factors affected the results, particularly in the case of my data set. When you have such a small sample, it's 21 women in the 2 BSO groups. Once you start breaking them down based on occupation or education level, we don't have a sufficient sample to run statistical analyses, but definitely that is something we need a larger data set to look at that, to see how those intersectional factors might affect the data.
So please feel free to ask, we still have more minutes with Dr. Almey. So if you have other questions that you would like to ask, feel free to do it either in French or in English.
I'm just going to launch a poll that we have just while we answer some more questions that we have. So if our participants can just take a couple of minutes to answer some of these questions, that would be greatly appreciated. And there's also going to be a survey launched once the webinar finishes as well. So I just have a question with respect to studying women and older adults. I'm just wondering if you could answer why it is that we should continue to pay closer attention to women's health, in particular as it relates to dementia, and also maybe just commenting on how historically a lot of research has been conducted on people who are older, such as 65 plus, but your research looked at aging with what we could call younger adults. So I'm just wondering if you could comment a little bit on those two things.
Yeah, so I think along with intersectionality, so great previous question, precision medicine is really becoming, I guess that kind of speaks to intersectionality, is really becoming a hot topic because there's a lot of evidence that we are better able to treat people when we understand sort of more nuanced things about participants. And sex differences are just one of those things. So if we are able to understand Alzheimer's disease in women and men specifically, it definitely has the potential to let us intervene, hopefully before they start to get symptoms of Alzheimer's disease. So more specific interventions based on For example, men, although men do have a decline in testicular hormones, they don't have the same sharp decline as women. So it sort of peters out in men over a 20 or 30-year time and really drops off in women within a few years. So we think maybe that time in women when they lose their hormones, whether that's following surgery or following natural menopause, might be a risk period. And if we can intervene then, we might have benefits for women. And that doesn't apply to men because they don't have that same sharp decline in hormones that might be supporting cognition. So better understanding specifics of diseases and disorders like Alzheimer's might make us better able to treat everyone, like women, but also men. If we understand what aspects of Alzheimer's are specific to men, we could develop similar interventions for them. And I think really in looking at a younger age group, I know we're not the only group doing this. So the goal really, my understanding is that once the decline, once people are diagnosed as mildly or with mild cognitive impairment, the decline can sort of be decreased, but it's more challenging. So there's a real hope of intervening before major cognitive decline and cognitive declines in the brain begin. And to do that, we need to understand the very early stages, the prodromal stages of Alzheimer's disease, so that we can get those interventions in before Alzheimer's even really takes hold. So I think that's one of the goals of our research, to better understand the time course of change in these women in the hopes of intervening before major detrimental changes are occurring to decrease Alzheimer's risk.
You mentioned that the women who were involved in the study were at about 5 years after their surgery. Do you think, or do you know if there is a plan to look at similar research for BSO recipients with 10 or more years following their surgery?
I don't know if that is entirely in the cards for the Einstein Lab study. I think probably that's always the dream. And so the women in this study, actually, I took data points from the first time they did an MRI scan in the study and their corresponding cognitive data. They are seen 3 times with the goal of being once a year. Sometimes it's a year and a half, depending on if life gets busy for the participants. So the goal right now is to track them over a 3 to 5 year period. But I know that Dr. Einstein does talk about this group of women as a cohort of women, and there is the potential to recontact these women down the road. So I would say this sample of women might be the sort of group that if you stop seeing them after 3 years, you can reach out to them 10 years following that and see who's willing to come back in and get an idea, get some of that data that you're talking about, because that really would be interesting to look at the change across time in this group.
Another question that I would have, if I may, you mentioned in your results the impact on cognition and the cortex thickness. Have you seen the same results when it comes to sleep? Because you mentioned that those who had the BSO, they were sitting, with hormone therapy, were sitting in between the control group and those without hormone therapy. So do you see the same effect when it comes to sleep and probably also mood and stress, or is it a different result that you observe?
That I honestly can't tell you much about because the data is still being analyzed. So I know a postdoctoral fellow, maybe a research associate now at the University of Toronto, Dr. Nicole Gervais, is looking at that right now. But I don't know if we see that same sort of stepwise thing where we have BSO without hormone therapy, BSO with hormone therapy, and then controls. I'm not sure if we're observing that in the other variables that are being measured. It would be interesting, though.
Maybe we will take a last question. You spoke about also the ethnicity of the group involved in the study, but racialized women are diagnosed with breast cancer and dementia at a higher rates than non-racialized women. The intersection of the race, gender, and health are significant for the results of this research. Is there any plan to do another study with a more targeted approach on this way?
I don't think Dr. Einstein is intending to do that right now. So to say We definitely have people of mixed race in the samples, or maybe not of mixed race, of different racial backgrounds in the sample, but they are predominantly Caucasian. Part of the reason is the BRCA1 mutation is carried, is very common in the Ashkenazi Jewish population. So a number of the women we see do have Jewish background, and it's just because this genetic mutation is more common in that population of women, and maybe less common in other racial backgrounds, or it may be that it's not as well recognized in other racial backgrounds if doctors aren't looking for it, or if it's not something their healthcare practitioners focus on. But I definitely think it would be interesting, because it's true, they're at greater risk of Alzheimer's disease in general, definitely African-American women. So if these women have their ovaries removed, is that greater risk of ovarian removal magnified in them as well, is really something we need to be looking into.
Thank you so much, Anne. I know that we're almost done, so maybe we'll just do another round table and see if there's any other questions between us. I just wanted to ask you, Anne, as maybe a concluding question, what is the one key takeaway from your findings that you would like people to know about? I know you touch on it, but maybe we can just repeat some of that from your research.
Yeah, for sure. So I would say the takeaway would be that sort of, even when we sort of try to extract age from the question, and we are looking just at loss of ovarian hormones, it does seem that that is contributing to changes in the brain and changes in memory. And since that is the case, I think there's real impetus to look at if and when hormone replacement should be prescribed to women, because there's definitely some caveats. We have to make sure it's safe. We have to make sure it's prescribed via an appropriate method, appropriate dose, at an appropriate time in life. But our results would suggest that there might be benefits to that, and we really need to look into that to benefit all women, since at least in the Canadian cohort and a number of other areas of the world, women do still seem to be at higher risk for Alzheimer's disease.
That's great. Thank you so much. And I just wanted to point out here on our last slide that, as Caitlin had mentioned, the recording of Anne's presentation and this chat will be made available on our connection site for Alzheimer Society staff. And for anyone who's joining us that's not a staff member, you can reach out to your local society to get access to this information. And if you have any questions, you can always email us at psp@alzheimer.ca. And we just wanted to give a big thank you to Anne. This presentation has been really remarkable, and we're so pleased that we were able to help support this research. And also thank you to Noah for helping us today and facilitating the questions. That was great.
Well, and thank you very much for having me. It was really great to come and to talk to you guys about the work that I did. Really, as a thank you. It wouldn't have been possible without the funding from the Alzheimer Society. So you guys made this work possible, and I'm glad I was able to tell you a little bit about it.
That's great. All right, so I see we have a couple of thank yous coming in. And again, if anyone's looking for information, they can send us an email, but we're going to log off now. So on behalf of the Alzheimer Society, we're very pleased and grateful to you, Anne. Thank you so much.
Oh, yeah, no problem.
All right, thanks everyone. Have a great day.